Welcome back to the Classics of Immunology Journal Club. Before we get started, don't forget to like the video on the YouTube channel if you like it, and then also click on the bell, subscribe, check out my website, and particularly the writing tab on my website has all the classic papers listed there so that you can read them and decide for yourself as to whether what I'm telling you is correct or not. And today we are in 2008, and a paper that was published in PLOS One, the Public Library of Scientists One, in February of 08. And the authors are Zoran Popmihajlov and Kendall Smith, yours truly. The title is Negative Feedback Regulation of T cells via IL-2 and FOXP3 Reciprocity. And the background in this whole effort is as follows. And the background is quite extensive at this stage of the game, because it's really been going on since 1981. The background in terms of the authors here is Zoran was a postdoc who came from Serbia. And he was very, very good postdoc and very personable, number one. Number two, he was very meticulous and very careful. And so he did these experiments multiple times that we're going to talk about today. And the problem that we embraced in this particular paper is that you couldn't really. So the regulatory T cells were sold by their progenitors as suppressor cells. And in all the experiments that people brought to the fore, it was difficult. They could show suppression of proliferation of T cells, particularly when they added in very high concentrations of the so-called regulatory cells. But it was difficult to discern or to find out whether or not that you were dealing with passive suppression via the removal of interleukin-2. Because interleukin-2 would overcome the suppression. If you gave it exogenously and flooded the system, then there was no suppression. So was this just passive removal of interleukin-2, or was there active suppression in some way or another? And of course, the people who were really hot on regulatory T cells, suppressor cells, they wanted it. They didn't want to have to deal with this interleukin-2 stuff. They wanted it to be an active, magical, and mysterious thing that the suppressor cells did to the so-called responder cells. The story goes back in the introduction, and Zoran and I wrote this paper. So the introduction traces everything back to 1981, as I just mentioned. In the new book I'm writing, that's provisionally titled The Interleukin Revolution, I detail what happened in terms of after we worked out the interleukin-2 or T cell growth factor, the radio-labeled T cell growth factor binding assay to first define that the way that the interleukin-2 molecule interacted with the cells to affect their responses was by binding to very specific receptors that appeared on active T cell antigen activated T cells. And I in discussions with Alan, I said, well, this has all the characteristics of a classical endocrine hormonal receptor system. That's what we're dealing with. And Alan thought about that for a while, and he said, well, not quite. He said, if you can't demonstrate negative feedback regulation, downregulation, mediated by the hormone itself, you don't really have a classic hormonal system. So we did several approaches to that question at the time, but we finally ended up saying that, well, we can't really solve the question at this particular point in time. We need more assays for the positive effect of interleukin-2 on the cells so that we could find out whether or not there was a feedback loop going on and there was downward suppression on this whole system. And then it wasn't until 10 years later, 1991, and then 92, 93, 94, 95, that the IL-2 knockout mouse added some data into the system. And of course, as you know now, because we've reviewed the papers in our club, if you knock out IL-2 and it's not present during embryogenesis, the mice initially look fine. They are fertile. They produce babies. They are born. They grow up. For the first month, they look exactly like the littermate controls. But in the second month, they start to undergo a problem that looks like GVH disease. They become runted. They get skin lesions. They get hepatosplenomegaly. They get a lymphadenopathy. They are filled up more and more and more with activated T cells. And the IL-2 knockout mouse actually succumb. They die early in life, particularly when the knockout is put onto a BALB/C background. They start to die and are dead by 5 weeks after birth, which is, not much life in that kind of an instance. The question is, why is that? Everybody thought, well, the interleukin-2 is the T cell growth factor. They should just be immunodeficient. They shouldn't be having activation of the whole T cell compartment and systemic auto-inflammatory diseases. When the IL-2 knockout mouse was initially made by Ivan Horak and his lab over in Würzburg in Germany, I went over, and I think I related to this story before, and talked to him and so forth, and we decided to do some experiments together. And one of the questions that came up right away is that, well, if we give them IL-2, if we inject IL-2 into these IL-2 knockout mice from the time of birth, can we prevent this disease? And the answer to that question was yes. We published a paper in 1996 showing that. And the thing is that we could, as long as we gave them IL-2 on a daily basis, they didn't get the autoimmune disease. But as soon as we stopped, almost to the day, 3 weeks later, they started to show signs of runting and failure to thrive, and ultimately succumbed to the autoinflammatory syndrome. Simultaneously with this phenomenon going on with the IL-2 knockout mouse, the scurfy mouse was shown, which had been around since 1949, 1959, and underwent the same runting kind of syndrome and disease. Scurfy mouse was found to be due to an activation of the T cell compartment with lots of cytokine producing T cells around and so forth. And that was a phenomenon that was followed up and defined as that was the reason for the problem. Then along came Sakaguchi in 1995, showing that a very good marker for these so-called regulatory T cells was the IL-2 receptor alpha chain. That was 1995, and that really caused an explosion of work in the field. Everybody, because now they could define a subset of T cells that had these regulatory phenomenon. And then along came Ethan Shivak in 1998, who devised an in vitro assay for the regulatory T cells. And he did so by putting T cells, responding T cells, in vitro in the presence of antigen-presenting cells. And then he would add in increasing doses of the regulatory T cells, and the regulatory T cells would suppress the replication, the proliferation of the responding T cells. And he interpreted his experiments by saying that, and he showed that you could overcome this suppression by adding IL-2. And so he then assumed that, or interpreted these results by saying that, well, there was obviously a deficiency of IL-2 in the system, and that was because the regulatory T cells were suppressing the production of interleukin-2 by the responding T cells. And so the problem with that is that because IL-2 was driving the proliferation of the responding T cells, because you could flood the system with IL-2 and overcome this so-called suppression, you couldn't really distinguish between passive removal binding, internalization, consumption, degradation of the interleukin-2 by these increasing concentrations of regulatory T cells that were added into these in vitro experiments. The other thing that Shivak had said is that the suppression was dependent on the presence of antigen-presenting cells, macrophages. And if you took them out of the system and used purified T cells as responding cells, then you couldn't show suppression. So he said, ergo, the regulatory T cells are blocking the help from the accessory cells, helping the responding cells to make IL-2. Then along came FOXP3, 2001. And then in 2003, 2001 was defined at the genetic level. 2003, it was shown to be FOXP3 was deficient in Sakaguchi's neonatally thymectomized mice and in nude mice and so forth. And there was another then explosion of activity in the field. And everybody said, this is so wonderful. Now we finally understand the scurfy mouse and the X-linked disease, because FOXP3 was a X-linked gene and so forth. Oh, now then the only problem was, as this was, a lot of this work was done in the mouse. When people started working in the human or did work in the human system, it wasn't as clear cut. It wasn't black and white. And it was shown early on, but that if you activated human peripheral blood mononuclear cells that have T cells and B cells and monocyte macrophages and the whole ball of wax and NK cells, you could make FOXP3 negative cells become FOXP3 positive cells. It was also shown, the other thing that was thought was that in the mouse experiments, this was only in the helper cell compartment, only the CD4 positive T cell compartment. But in the human, it was shown, and Sakaguchi had said this early on, the smaller fraction of CD8 cells would become FOXP3 positive. And therefore, this was the sine qua non of a regulatory T cell, the FOXP3. So things got to be very murky around this time in 2006, 2007, 2008. So we had stayed out of the business. I had stayed out of the business of these regulatory T cells. And I was watching it carefully. And this was the first time that we decided to get involved. This paper that we're going to review today. Along the same time, and we reviewed this paper last week, Pushpa Pandian and Mike Leonardo from the NIAID and the NIH had shown very, very nicely that the regulatory T cells and the FOXP3 positive cells actually consumed IL-2 and led to cytokine deprivation apoptosis of the responder cells. So I was sort of biased, and I liked that. I liked that because that was what I had said all along from the very, very beginning of this whole saga. So we decided that around 2006 and 7 and so forth to get involved and to do some experiments. And we focused on the human system. And we wanted to know what was going on with FOXP3 expression by human T cells after T cell receptor activation. And we wanted to know what's going on in terms of, is it FOXP3 that's really suppressing other cells, FOXP3 positive cells suppressing the responder cells, or exactly what's happening? Let's go to screen sharing and look at the results. This is figure 1. And in the A portion in the upper left, are just freshly isolated cells, and these plots of flow cytometry plots are plots of isotype control antibody versus antibodies to FOXP3 versus CD25, and you can see that freshly isolated cells are for the most part FOXP3 negative. There's a small fraction here of cells that are the highest density of CD25 that seem to be FOXP3 positive in the CD4 compartment. And in the CD8 compartment, it's very, very small. Now, Zoran did 19 different experiments and found out essentially exactly the same thing. This is the results of 4 experiments where he compared after activation through the anti-CD3 in PBMCs, and you can see up in the upper portion of the B figure are CD4 cells, and in the lower is CD8 positive cells. This is time in hours, going over from time 0 up to 150 hours, 5 days essentially. And you can see in the open symbols is CD25 expression on these cells, and in the solid symbols is FOXP3. There's a different scale. This is CD25 scale over here. This is FOXP3 over here. And you can see the first thing that happens is within 12 hours, you see CD25 positive cells, but the FOXP3 is essentially unchanged from time 0. At 24 hours, you've got almost maximal CD25 expression, and it isn't until 48 hours then, or no, it isn't until 24 hours that you see peak FOXP3 expression. Then in the C portion of this, is to emphasize is that in these activated cells, the FOXP3 expression is really only in the highest density of alpha chain positive cells, both in the CD4 as well as the CD8 compartment. And here is in histogram plots of, this is the amount of FOXP3 expression in CD4 positive cells in the solid bar, and in the gray bar is CD8 positive cells. The FOXP3 expression goes up 5-fold in the CD4 compartment, and it goes up 50-fold in the CD8 compartment. So what are these FOXP3 expression was supposed to be the sine qua non of a regulatory cell. And so in figure 2, the question was, so well, if the interleukin-2, and this has been shown by others at this point in time, if interleukin-2 stimulates FOXP3 expression, and what happens if you add interleukin-2 at time 0? And here we've got a plot of FOXP3 on the ordinate and hours after anti-CD3 activation. And what we have here is when we supplement interleukin-2 at time 0, and then we look at FOXP3 expression, we don't find the FOXP3 expression coming up earlier. And this, in the early stages between 0 and 12 hours, we see it coming up by 24 hours. And then IL-2 supplementation prolongs the expression of FOXP3. If you look at the alpha chain positivity, it comes up in both compartments and stays up essentially over the course of the experiments, both in CD4s as well as CD8s. And you see the same phenomenon of interleukin-2 prolonging the expression of FOXP3. So then if you move on to figure 3, can you show any difference in terms of what happens if you try to compete away the IL-2 receptor interaction? And so here's an experiment. What we're looking at here is FOXP3 expression without any interleukin-2 or in the medium. You get this amount of FOXP3. If you add in interleukin-2, it goes up a little bit. If you then take anti-alpha chain positive with increasing concentrations of anti-receptor, IL-2 receptor, you see progressively decreased expression of FOXP3. And then you can overcome that suppression by adding back in interleukin-2. So then if you look at the effect of anti-interleukin-2, you see a progressive decrease of FOXP3 expression in the presence of increasing concentrations of IL-2 antibody, and then you can overcome that suppression by adding in interleukin-2. So there's a reciprocal relationship between these two things, and which stimulated us to go. We're going on to figure 5 now in the paper. I forgot to say that before we did these last experiments, we found that other gamma chain receptors and other gamma chain cytokines, interleukin-4, interleukin-7, interleukin-15, interleukin-21, they were all negative. It was only interleukin-2 that would do that. The next thing we did was an experiment or a kind of an experiment that nobody else in the business had done over the course of this entire time period. And that is to stain in the same cell population FOXP3 and interleukin-2, because the idea is that furthered by or proposed by Ethan Shivak is that the FOXP3 positive cells suppressed interleukin-2 production by the FOXP3 negative cells. And so we took CD4 positive cells, we looked at them 0 hours, 24, 48 hours, and so forth. And what we have here is FOXP3 on the ordinate again, and now we have interleukin-2 on the abscissa. And so what you see here, and at the same time we stained these cells before the experiment started with a CFSE to track the divisions of these T cells. So this is time 0, and there is just a single component, meaning that they have not undergone any division. And then at 24 hours, still there's no division. But now we see 8% of the cells are FOXP3 positive versus 3.4% before. And now we see a little bit of the cells going that are becoming to be IL-2 positive. And then if we then go to 48 hours, they still have not undergone a cell division, but we have 10% FOXP3 positive cells and 2.2% IL-2 positive cells. By 72 hours, FOXP3 is 6.4, and now we have 4.8% IL-2 positive cells. And by 96 hours, 4 days, we have a decrease in IL-2. But between 48 hours and 72, we've undergone 1 division, and here's the beginning of a 2nd division. And so we have, by 96, you can see that the cell population has started to proliferate. Now, so we wondered, so the thing is that there's a reciprocal expression of FOXP3 versus IL-2. So FOXP3 expression is inhibiting, perhaps, the gene expression of interleukin-2 in that population, but it doesn't look like it's doing anything at all into the FOXP3 negative population. So we thought, well, let's re-stimulate these cells. And here we are. We still have, what we did was we started with these cells at division 0, and we've looked at, we have 59% of the cells at division 0 at 72 hours. And then here we have 0.8, or no, wait. I'm sorry, 29% in the first division. And we have FOXP3 here, and we have, for the most part, very little IL-2 positive cells. They're in the FOXP3 negative compartment. So that we didn't really have evidence that we have suppression of this population by this population. So then we thought, well, let's re-stimulate these cells. So we cultured the cells for 24 hours, washed them free of all their activation and so forth, and then re-stimulated them through anti-CD3 and anti-CD28, and looked at the whole thing again with FOXP3 versus IL-2, and we find that exactly the same story with a reciprocal expression of FOXP3 and interleukin-2. So we found So we really found no evidence that the FOXP3 positive cells were suppressing IL-2 production by FOXP3 negative cells. So let's stop the screen share and go on to the discussion. So that's it. The conclusions that we enunciate in a pretty long discussion here was that the IL-2 receptor interaction is responsible for the increase in FOXP3 expression by anti-CD3 TCR-stimulated human T cells, what was already known, essentially. But that led us and our data in this particular paper to conclude that there was an IL-2 FOXP3 negative feedback loop, exactly what Alan Monk, my endocrinological colleague, wanted me to demonstrate to him before I could start calling this whole system a hormonal system. And that turns down the activation, the interleukin-2 receptor interaction positive stimulation of the cells. And so that really reinforces the idea that if the cell becomes FOXP3 positive, then FOXP3 in a transcriptional, because it's a transcriptional activator and repressor, and other people had shown by then that one of the things that FOXP3 does to turn down cytokine gene expression of IL-2 and other cytokines as well is to bind and prevent NFAT from interacting at the promoter region of the cytokine genes, and also it binds to and prevents an NF-kappa B participation in the activation phase of this whole thing. So that's what our data show is that would be anticipated by that whole phenomenon. At the same time, and we reviewed this paper too, back in one of the other journal clubs, Jerry Ritz's group had shown that FOXP3 is a STAT5 induced gene. It's an IL-2 induced gene. So all of these results are consistent with the primary mode of action of these FOXP3 CD25 or alpha chain positive regulatory T cells is really as a passive sink, removing the interleukin-2, which is really the positive growth factor in the whole T cell compartment. And we could find no evidence that there was any suppressor phenomenon going on of the FOXP3 positive cells interacting with and suppressing IL-2 gene expression by FOXP3 negative cells. So that's where we left it in 2008. And now it's 2022. So we're 14 years later, and other things have happened in the meantime, in the past few years. One of the things that happened was that Shimon Sakaguchi, with a new postdoc from the Karolinska Institute from Sweden, Kaya Wing, published a paper that came out in Science in 2008, looking at the role of CTLA4 in Treg-mediated suppressive phenomenon. And we'll review that paper next. And that was followed by another paper from 2010 by Gregoire Altan-Bonnet and myself. And that paper just reinforced Pushpapandian's results, showing that there was a fantastic mechanism whereby Treg cells could sop up IL-2 very rapidly. Then in 2016, Kaya Wing, having gone back to Karolinska after her postdoc with Sakaguchi, looked at an induced CTLA4 knockout mouse to see what would happen if she took out CTLA4 after embryogenesis and after initial development of the immune system. So stay tuned. We're going to review those papers too. So before I go, don't forget to like the video and click on the bell and subscribe and check out the website. And thanks for coming, and come back next week. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.